ठीक है ना, चालू कर चल। इनपुट बंद मत कर ना, तेरा इनपुट चालू कर। इनपुट कैप्चर तेरे को लगेगा।
ये माइक एक्सपोजर
अरे लेकिन
सेम चीज है।
कोई चीज सेटिंग मत चेंज कर, तू पहले आवाज आ रहा था। अच्छा, हम लोग ऑलरेडी लेट है। कैमरा किधर है? कैमरा किधर है?
कैमरा नहीं, कैमरा लगाने को नहीं बोला।
चल, स्ट्रीम चालू किया?
हाँ, स्ट्रीम चालू है।
शुरू करें हम लोग।
स्पीकर को पास ही रखना, नोबडी टॉक्स दैट। ये भी है यानी कहने का सीरियस टू तो बता रहा हूं, सारे क्या नहीं बड़ा मतलब है वो अंदर में थोड़ा कुछ
A very warm welcome to everyone present here for ANVESHA 2022, the official science fest of IISER. We would like to start with the Nobel lecture series. First up, I'd like to call Dr. Nishana, Assistant Professor Grade 1, Biology and Physics. Dr. Nishana is a vivacious young scientist exploring the profoundness of nuclear organization, chromatin architecture, genomic instability, and cancer biology. She completed her PhD in biochemistry under Dr. Satish C. Raghav, Department of Biochemistry, Indian Institute of Science, Bangalore, and proceeded with her postdoctoral research at laboratory of Professor James Stock, Department of Pathology, New York University, Langone Medical Center. She is also the recipient of Aggressive Cancer Research Grant from the National Cancer Center and was selected as the best research presentation at the Postdoc Research Symposium of New York University Langone Medical Center. Dr. Nishana, you will be glad to have you. Can you hear me? Yes. Can I open it from the beginning or is it straight? Oh, okay. So slide change. Oh. Do I have a laser or slide changer? Okay, so yes. Yes. Slide changer. Yes. Yes. Thank you all. So today I'll be speaking about Svante Pavo, how his research helped in unrevealing the caveman within us. Svante Pavo, unveiling the caveman within us. Svante Pavo got the Nobel Prize in Physiology or Medicine in 2022. He was born in Sweden in 1955, and through my slides, I'm going to tell you about how he had a strong passion to do research in fossils and mummies, and how every time he had a chance to deviate from the topic, he came back, and how he used his expertise that he gained otherwise in order to solve the questions pertaining to fossils and mummies. The prize that he got was for his discoveries concerning the genomes of extinct hominins and human evolution, and this Nobel Prize was shared with none in 2022. So, Pego had an intense passion towards studying fossils. He had a passion towards visiting pyramids. And when he was 13 years old, his mom actually took him for a vacation to Egypt. And that was the time when he decided that he wanted to pursue research on mummies. And his dream was to become an Egyptologist, to go to Egypt, to excavate the pyramids, dig out the fossils, and study the genomes of these fossils. And because of this desire, when he was 20, in 1975, he started at the Uppsala University on a course named History of Science and Egyptology. So his dream, as I said, was to study the fossils, the mummies, but then what happened eventually was he was not really visiting any of these pyramids. He was stuck to a library in the Uppsala University and he was digging through books, learning about the language, the structure, these writings on the walls of the pyramid. So obviously he was not happy. He quit it in 2 years and then he took up a career in medicine. And this actually in turn was inspired by his father. His father was again a Nobel Prize winner in biochemistry for discovering prostaglandins. And inspired by his father, Pablo wanted to do research in, and Pablo wanted to pursue medicine and get a job. That was what he told. He thought that he would get a profession by learning medicine, and he joined into medicine. But within a few years, he realized that again, medicine is not his passion. In fact, he really enjoyed doing medicine, but his prime interest was again research. So he switched back to research and joined the lab of P.A. Peterson in 1981, and that was to study adenovirus and their interaction with immune system. Nothing to do with fossils, completely on molecular biology and cell biology techniques, and he did extremely good in his PhD. These are a few screenshots of his papers during the PhD. This is just a few of them. There were others more. Despite doing really good in biology, publishing in Cell, he was not happy. He had the passion of fossils with him. He had to work on mummies, he had to work on pyramids. He's, he then decided that he would use all the expertise that he gained during his PhD to go back and study fossils. So, during his PhD, he gained expertise in all of these molecular biology techniques about how to study DNA. And the question he had in mind is, why can't I use this expertise in studying DNA of mummies? So did he ask permission to his PA, I mean PI? No, he didn't. He secretly did his research during the nights and during the weekends without his PI knowing on fossils. And this he did with the help of his previous colleagues at Uppsala University, mentors at Uppsala University. So he basically convinced them to get him a piece of fossils. This is what it is. I mean, one of his former Egyptology professor, he took fossil remnants from German museum and tried isolating DNA from that. It was not just one or two samples, he took 23 samples from the museum and started digging out DNA from that. And this, again, was without the knowledge of his PI. Did he succeed? Yes. He did succeed. And in 1984, he published his findings in a very small German journal, which says whatever in German. It says that he was able to detect ancient DNA from samples from the museum in Germany. So that was a small paper, but it was a big hit. In Nature, 1985, he published his finding on molecular cloning of ancient Egyptian mummy DNA, and that actually was covered in the News and Views of Nature, Mummified human DNA cloned. While he was writing this, actually, and see, there are a lot of difficulties in doing this. It might seem easy to us initially, but the thing is, see, these samples were preserved several years ago. They might be intact, they might not be intact. They would have acquired DNA damages throughout. So getting these DNA is really very difficult. So this was the time, we should go back to the times of 1980s when there was no PCR. Polymerase chain reaction was non-existent. So what he would do is he would isolate the DNA, shear it into small pieces, clone it into bacteria, and then sequence it. So the paper only had a sequencing component in it, and see, it was a nature paper. Remember, this is 1984, 1985. So while he was doing this, fortunately or unfortunately, he was cooked. Okay, so this is a glimpse into the first paper. What you see here is a tissue section of the skin from the lower leg of a mummy. And these mummies, the 23 mummy samples came from 2370 to 2160 before Christ. DNA libraries were cloned into bacteria. Even though he used these 23 samples, only one of them was successful. And this one clone came from a child who was less than 1 year, and radiocarbon dating showed that the sample is 2430 years old. So this one sample he was able to sequence, and it was a Nature paper which came in 1985. While he was writing this paper, he was kind of scooped, not with human DNA, but another DNA, quagga. So this is a zebra-like animal which was extinct, and from the lab of Alan C. Wilson, they published again in Nature the sequencing of samples from this ancient zebra-like or horse-like animal. And this actually became the first paper on sequencing of ancient DNA. Paper was disappointed that he was kind of scooped. But again, he was someone who, when life gives you lemons, make whatever. So, what did he do? This was again before the internet. He wrote a mail. So I put the picture of the air mail just because I know that when I say mail, we'll automatically take it to be an email. He wrote a mail to Wilson, and in this mail, he attached the proof of his paper, the Nature paper, which is going to be published in 1985. He sent it to Wilson and asked his opinion on this paper. And Wilson replied back asking if Wilson can join Pabo's lab for a sabbatical. Sabbatical is basically like a project internship which PIs do. Now, Wilson had no idea that Pabo was a grad student. Wilson had no idea that Pabo didn't have a lab or didn't even have a PhD. He assumed that Pabo was a good, big scientist, and Wilson asked him if he can do sabbatical in Pabo's lab. And Wilson actually was a very prominent geneticist at that time. Pablo was perplexed and he replied to him telling that, see, I don't have a lab, but if you would let me, I would like to do postdoc in your lab. Wilson was happy. And this team, this combination was the right place, right time, and right team. You know why? Because, see, Wilson's team they already have an experience in isolating ancient DNA. Well and good. Technically, they're nice. Second, this was the time when PCR was introduced. Carimulis, he had just invented the PCR technique, and Wilson's lab was the first lab outside CETUS. CETUS was a company that had the patent to use PCR at that time, and Wilson's lab was the first lab which could use PCR outside CETUS. So, this was the right place for the Pabo now. He could, instead of cloning it, amplify the DNA and then sequence it. Very easy, right? Ancient DNA, Wilson's lab has all the techniques necessary, all the equipment necessary, do it, finish it, publish. Is it easy? It seems easy, but it's not. It was not easy as it sounds because this DNA is ancient DNA. They have been preserved since millions.